The overall goal of this procedure is to determine the polar lipid composition of Arabidopsis seedlings. This is accomplished by first extracting lipids from the seedlings. The polar lipids are then separated by thin layer chromatography. Next, specific lipid spots are isolated from the thin layer plate and converted to fatty acyl methyl esters by transesterification. The final step of the procedure is to quantify the composition of fatty acyl methyl esters by gas-liquid chromatography. Ultimately, results can be obtained that show the molar ratio and the fatty acid profile of each polar lipid in wild type and mutant.
Hi, my name is Christoph. Many of you have asked us how to do a quantitative analysis of polar lipids. We use a very simple and robust method that served us well during large-scale screening of mutants in the model plant Arabidopsis or the algae Chlamydomonas. Other excellent methods such as mass spectrometry-based lipid profiling require expensive equipment and lots of experience and training. We use a two-stage procedure that involves simple thin-layer chromatography combined with quantitation of fatty acid methylesters. Following the isolation of mutants, usually a more quantitative analysis is required. This is done by isolation of membrane lipids from the thin layer plate and preparation of fatty acid methylesters, which are quantified by liquid-gas chromatography. The method is highly reproducible and results are comparable with more complex mass spectrometry-based techniques. Demonstrating the procedure will be Zhen Wang, a graduate student from my laboratory.
Begin lipid extraction by harvesting 30 mg of 4-week-old Arabidopsis leaves.